So this is our next topic, which I believe is covered in Chapter 3 of your book. So for microbiologists like myself to be able to see a lot of the microbes that we work with in the lab under the microscope, these organisms don't have natural color to them. They're relatively translucent. If you were to just slap them on a slide and throw them under the microscope, you're not going to see them. So a lot of times we stain them. This creates contrast for us to be able to visualize them. But we can go one step further with staining. We have lots of specialized staining techniques out there that allow us to not just see what we want to look at, but also tell us a little bit more information about that organism based on how it's stained. So differential staining, particularly the Gram stain, is a very popular type of staining technique. So a differential stain, as the name implies, differential, right? What is it allowing us to do? See differences between organisms, right? In the case of gram-positive and gram-negative, organisms based on their cellular structure, and anyone know what cellular structure is different that's going to cause us to get different colors in this? Peptidoglycan, right? That cell wall component found only in bacteria is what helps determine whether when you stain a bacteria, whether it comes out purple or pink. So differential means that you're going to differentiate one group from another based on color. But that color, of course, is determined by the differences in that cell. So in the case of the Gram stain, the difference is that cell wall, right? How much peptidoglycan is there in that cell wall? How much of that substance that makes up bacteria's cell walls is present? So the steps for the Gram stain, and of course those you guys in lab would do it, right? And I also posted for you guys, I think I posted already some videos of me actually doing it in lab. And again, I post those videos, those animations and stuff to help you guys, right? Some of us are visual learners, that's really helpful. If you feel like that's just a waste of your time, then fine, skip it, right? It's just there to help you, right? And I only put, stuff that I know is helpful. I don't try to overload you guys, right? Too much stuff. So what you do need to know is the steps of the staining procedure and the basic principle behind how it works, right? You don't actually have to do it. Someday you might, right? Someday you might. So the first step is applying the first stain in the procedure, what we call the primary stain. In this case is crystal violet. The next step is what's called a mordulant, and the reason for that is it adheres to the crystal violet, and that is called iodine. And it's straight up iodine like you get in your medicine cabinet. Notice the cells all of them get stained purple, and then they're going to remain purple, right? The iodine doesn't change the color of the cells. If you were to stop and look at them under a microscope, they'd all be purple. What makes a differential stain differential is that all those differential stains are going to have some type of decolorization step, where you're going to remove the color from one group of organisms, but the other set of organisms are going to retain that color. So in this case, the ones that retain the color, we call them gram-positive. So they're going to stay purple. Positive and negative does not have anything to do with charge here. It has to do with the result in a test you're running, right? You're running a test. You're doing a Gram stain. Okay, do they stay purple? They're positive, right? So I always remember P for positive. Purple for positive. The ones you decolorize, you're not going to see them if you stop there. They become colorless. Right? We use ethanol in the lab. In the past, some people have used acetone. Acetone is a really good decolorizer. Those of us that get our nails done, we know all about acetone. Right? It smells really bad and it is extremely damaging to mucous membranes. It's a very strong irritant, which is why we don't use it in the lab, why we prefer to use ethanol alcohol because it works just as good in this procedure as acetone does. Unfortunately, not the case when it comes to nail polish. They have alternatives out there. I don't know what they're even made of, but good old acetone is always the one that works best. But you can visualize that, right? Removing that color, right? We're using some type of reagent that's going to help remove color. The same is true, right, if you accidentally write with Sharpie marker on our wiper boards or any slick surface. Permanent marker, Sharpie markers aren't truly permanent. Most cases, you can wipe it off with alcohol. But there are some Sharpie markers, industrial strength, that you can buy that are ethanol resistant. And so if someone writes on your board with that, you're kind of screwed. But we use that, you know, in the lab sometimes because we don't want it to wash off with ethanol what we wrote. So we remove that color, but we want to see those ones that we remove the color from, right? So we have to do one more step. We have to do what's called counter staining. In this case, we use safranin, which is a red dye. But because these cells have been damaged in the process, they don't actually appear red under the microscope. They're going to be a lighter shade of red, known as pink, right? And it's important in this order, right, that we start with that dark, vibrant color, purple, crystal violet first, and we follow up with the red saffron dye, because otherwise we wouldn't be able to distinguish the two. Because guess what? Those purple ones, do they get stained with the safranin? They do. They do. But because they were purple to start with, when you stain them with that red, they still look purple. So I always use the example of laundry. Most people understand that whole problem with laundry, right? So I grew up in a relatively large household, right? So we could separate into literally the different colors. We had the whites, we had the darks, we had the lights, lighter colors, right? And so one day I put all the whites in, and accidentally a red sock got in there. So my dad's underwear were pink for a while. Yeah, he was sort of okay with that, right? Because it ran, right, in the cycle and made everything pink. My current husband, I love him very much because we can throw everything in together. Nowadays, a lot of that stuff you don't have to worry about, right? That, and he has black underwear. There's no way I could screw those babies up, right? Even if it bleeds over, they're not going to change from being black, right? If red runs onto it or purple runs onto it, you're not going to notice, right? Makes sense? If anything, they fade over time. Okay? So being purple and then getting stained red, again, you're only going to see purple because it's a much darker color. Right? You can't flip this order because if you did red first and then purple, everything's going to be purple. You're not going to tell. You had to tell light purple from dark purple. That wouldn't be very fun, right? I'm telling you, even the pink and purple sometimes is hard to see. Okay, so this one, I don't know if I had posted it for you guys. This is a newer animation website that a colleague had shared with me. And I really like the visuals that it has. Oh. What do we not have today, y'all? Okay, so we will table that one for next time. Moving on. So I had posted the gram stain animation for you guys. I have this other one. I do want to go through it step by step for you guys. And go down, we're going to go down one more level, right? We're going to talk about what's happening. Okay, so purple's, the cells are getting stained purple. Why are they staying purple? Why are they not staying purple? What's the difference between these gram-positive versus these gram-negative cells? And unfortunately, I don't have the tools I want today. So we will come back to that. Yeah, it's still flashing and not green. So this table, right, goes over each one of those steps, right, down to the molecular level, that level of understanding that you guys need to do. But I really want my visual aid, so we're going to skip over, and these are visual aids as well. So we'll come back to that. And that'll give you guys who haven't watched that animation yet more time. Not the case for anybody? Good. All right. So where is the cell wall for bacteria, right? What is it made of? What is its chemistry? What is peptidoglycan, right? So we're going to dive in deeper, right? What is this stuff? And, some cells have additional components. They have tectonic acid, teichoic acid, excuse me. I added another T for some reason. Some of them have an outer membrane that we're going to see next time. Some have lipopolysaccharide. Some have porin proteins and a periplasm. And again, some gram-positives have some of these structures. Some gram-negatives have these structures. So who's got what, right, is the question here. And helps us distinguish between these two different cell types. So for the bacterial cell wall, so if you have a cell, you have what's that goo in the middle? What do we call that? Cytoplasm, cytosol, right? It's mostly aqueous. It's got all the organelles in it if you're talking about a eukaryote, right? If you're talking about a prokaryote, we still got ribosomes. We still got to make proteins. You still got DNA, right? We just don't have lots of membranes inside, but we definitely have what? Defining that cell. Before that, do we have cell walls? No. What makes our cells a cell? The cell membrane, which also has a gazillion other names, right? Cytoplasmic membrane, phospholipid bilayer. I'll stick to cell membrane, okay? Always. But know that those other names exist out there, right? Cytoplasmic membrane, plasma membrane. So if you're a cell that has a cell wall, it's outside of that membrane, that cellular membrane, that membrane that defines the cell. And it is rigid. Unlike membranes we know are what? Are they rigid? No, they're flexible, they're dynamic, they move, they change. Especially when you visualize that amoeba, right? Boy, he can change the shape of that membrane. But cells that have cell walls, on the other hand, do they change in shape? No, because what did we just say about that wall? It's what? It's rigid, right? It's literally like the walls you look at surrounding this room. But different from this room, and is that wall like continuous, right? Is it no holes in it at all? Is it nice, smooth surface? There's no way we're walking through this wall, right? None of us have superpowers. Shoot. Okay. So, but if a cell was like that, if their wall was a wall like the wall surrounding this room, would they be able to survive and exist? No, because stuff has to what? Get in and out, right? So it does have openings. It does have doors. It does have windows, right? There is escape. There is entrance. In order for stuff to move in and out. But the rigidity helps hold and maintain its structure. So think again like insects, right? They have an exoskeleton. You can almost say that part of their skeleton for cells that have a cell wall is that wall. Helps maintain their shape. But another problem for especially single-celled organisms is the pressure of the world on them. Particularly the pressure that water can create, what we call osmotic pressure, right? So when they're in the environment, remember, big holes in that wall. The water can come rushing in. What happens when it rushes in, it's going to start swelling, right? What happens to our cells if we put it in an environment where there's too much water? They burst. So bacteria, they're a single-celled organism. They can't afford to lose a cell. They are a cell, right? If they burst, they're what? Dead, right? So they have to have a way of protecting themselves from this problem of osmotic pressure. And that's what the cell wall does, right? That's the main function of their cell wall, is help them to resist osmotic pressure, keep them from bursting. As a side, it helps form shape, right, and structure for them. This cell wall is unique among bacteria. We can divide them into two categories, gram-positive versus gram-negative, based on how they stain. But again, how they stain is related to what are they made out of, right? How does that cell wall interact with the dyes and the decolorizers and such that we use? I have no watch today. Okay, I have to keep turning the computer behind me on, so I have a No watch, the watch battery died, and forgot the cell phone at home. Then no internet. It's like one of those days where you want to go back to bed and start all over again. Ah! The good news is I could do this stuff in my sleep. So sometimes you'll see it abbreviated PTG. Again, this substance is called peptidoglycan. It makes up the cell wall of bacteria only. Right? Only bacteria have got this. What is it made out of? It is made out of sugar, hence the glycan in its name. That refers to the sugar component. It's actually two modified sugars, N-acetylglucosamine. N-acetylglucosamine, commonly abbreviated NAG, N-acetylmuric acid, commonly abbreviated NAM. The good news for you guys is you don't have to spell them or say them. You have to be able to recognize them. These are alternating, which means you have one and then the next, right? So imagine, right? I should have brought in my alternating Mardi Gras beads today, right? Alternating colors. Alternating sugars in a string. But this is a three-dimensional string. So imagine a jawbreaker, right? Covered in layers of sugar, right? So you have this mesh network of alternating sugars completely covering the surface of the cell. And that's just one layer. Then you have another layer of sugar and another layer. Layer upon layer upon layer. As many as 30 layers for a gram-positive organism. But when we stack layers, right, we start to run into a problem. So imagine if I got up and stacked a whole bunch of bricks in front of us, right? Would that be a very stable structure if I just stacked them up row after row after row after row? No. You could walk up and knock it over, right? So what do we put between the bricks to help hold them together? Mortar, right? What do we put between the sugars to hold the layers together? The sugars of the glycan chain, right? We string them together with 4 amino acids. That's why it's commonly referred to as a tetrapeptide chain, because tetra means 4. Peptide refers to the bond between amino acids, hence where we get the word peptidoglycan. It's a little bit of amino acid and a whole lot of sugar. So this is a picture of it, but we're going to zoom in, right? My next slide zooms in. So again, these are sugars, right? Here are the 4 amino acids. For gram-positives, because I said they could have as many as like 30 layers of sugar, they have an additional bridging here. These additional peptides, these additional amino acids that help hold these two linkages together. So that's their wall, and again, you know, there's holes in this, right? Stuff can get through. Think of it as a wicker basket, right? We can put our Easter eggs in it coming up, but you certainly wouldn't put water in it. Okay? There's holes in it. It's rigid. It can hold the structure, right? It can hold the big stuff in, but the little stuff just goes whoop, right through. Same is true when we start dyeing these cells, right? When we add stains, it can go right into the cell, right? Right through that cell wall. So as I said, gram-positives, very thick layer. You know, as many as 30 layers of that sugar. Regardless of how thick it is, that peptidoglycan is permeable. Stuff goes through it. It has to, otherwise the cell wouldn't be able to survive. In addition, gram-positives associated, and you can almost think of this as rebar, right? When they're getting ready to, and I know in my neighborhood they're like destroying all my sidewalks. They're replacing the gas lines. It's a nightmare, right? When I see the rebar going down, I'm like, yay, they're going to pour the cement. I can get my sidewalk back, right? Because that helps stabilize, right, that substance. Same is true for the teichoic acid. These are acids. These are substances that run through the layers of peptidoglycan and kind of stick out even, right, and help add additional support to those multiple layers of sugar and amino acid, that peptidoglycan layer of the cell wall. So I should have a picture, yeah. Okay. So you see the red lines, right? So there's our rebar running through to help stabilize this very thick structure of peptidoglycan. So when you look at these cells in cross-section, right, all cells are going to have a cell membrane, cytoplasmic membrane. In the case of gram-positive, I said that peptidoglycan is going to be very thick. So you can even see it, right, this thick layer. This is an electron micrograph, which means an electron microscope did this. You're not going to see that level of detail, right, with our microscopes. You're lucky that you can see individual bacterial cells. Our resolution is about to that level, right? You're actually just seeing the cell, and even the cell itself is quite small. For gram-negatives, in contrast, they are much more complex than gram-positives. And notice the color scheme I've got going here, right, to help you guys remember. Gram-positive are purple, gram-negative are going to be like this pinkish color when we stain them. If they contain it at all, they contain a very thin layer of peptidoglycan. And it's actually sandwiched between two membranes. There's cellular membrane and what we call the outer membrane. This is the outermost covering of that cell. That region between the two layers is referred to as the periplasm. And this is almost like an extra storage compartment for the cell. And it is typically considered outside of the cell, right? Once you pass the cell membrane, you're actually considered to be outside of that cell. So when we secrete things, they leave us, right? They leave the cell. So they go through the cell membrane, and they go into that space between the outer membrane and the cellular membrane. And in that space is a little bit of peptidoglycan, a little bit of rigidity and support. So when we look at it here, right, here's that cell membrane, your cell membrane, right, and then that space in between the periplasm space, and there's that little bit of peptidoglycan, right, in that space. And then you have this outer membrane, and this is a quite complex outer membrane. And in this electron micrograph, right, so you have the periplasm, the outer membrane, and then this organism even has an additional layer known as a capsule that we'll talk about soon. For this guy, when you look at this outer membrane, it doesn't look the same as the cellular membrane, does it? This top layer, or the outer leaflet, is made of a different substance. We know that to this substance to be called lipopolysaccharide. So what are membranes typically made of? What's that substance? You said it earlier. Phospholipids, right? So they're lipids with a phosphate group, that little circle denoted there. Here, we still got lipid component, right? Lipo. But now we've got polysaccharides in the equation. We've got some sugar, right? So here's the lipid component, the little square, and guess what these squiggly lines are? Poly means what? Many saccharide sugars. So they got a whole lot of sugar. And notice, because of that, this layer is kind of scary, huh? This is like trying to get through a jungle. Right? All these molecules, imagine all these sugars, right? Imagine if you were trying to get through there. Forget about it, you need a machete, right? So instead of a machete, what does this cell have to help move stuff through this outer membrane? It's got porin proteins. So these are very large protein structures that literally will open up holes in that outer membrane so stuff can flow through. Because again, stuff's got to get in, they got to eat somehow, and stuff's got to get out. Otherwise, you become toxic and die. So that lipopolysaccharide is of quite a bit of importance when as it relates to medicine, right? How we define disease, how these organisms affect us. Gram negatives, unfortunately, are really bad news for us because of this outer leaflet of their outer membrane, this substance we know as lipopolysaccharide. So it's still a bilayer, right? But the difference is that outer layer is a different substance, not phospholipids. It's lipopolysaccharide. So it is a lipid component with a whole bunch of sugar. And again, a very hard layer to get through. It's very protective for these organisms. And in order to still survive, though, they have to allow stuff in and out. So they have specialized porin proteins that do that, help them bring stuff in and out. When we start breaking down lipopolysaccharides, we can break them down into two major components of this molecule. The O-specific polysaccharide chain. So O actually stands for opposite, right? And so this is that very long chain of sugars sticking out into space. The lipid A component is that square block, that lipid component that kind of replaces what we're used to seeing, that phosphate group. And then, of course, again, there's the lipid component that comes down, the fatty acids, that interacts and helps form that bilayer of the outer membrane. So the O-specific, and again, remember, these are sticking out. All these sugars are sticking out, may even be the outermost covering of this bacteria. These can be quite distinct from one bacteria to another, literally down to different strains of E. coli. So when you hear E. coli 0157, right, when we've had an outbreak of this one, that can cause very severe diarrhea because this particular strain of E. coli carries the Shiga toxin. It's a toxin very similar to Shigella, right? Another bacteria out there that can cause really severe diarrhea to the level of what we refer to as dysentery, where it's literally destroying your intestines. You get bloody, horrible diarrhea from it. We know we can identify this strain by its sugars, right? So this particular strain that has these specific sugars that have been denoted O157 carries that toxin, that Shiga toxin, right? So scientists can use it to identify, yes, you've got this bad strain, although they'd pretty well be able to guess by your symptoms too. But you could have something else too, right? You could have Shigella and have that same problem. So, of course, we want to determine what you have so we can treat you best. So we can figure out what bad guy you have by determining what sugars that bad guy has. That helps determine it. The bad news with gram-negative bacteria, okay, this sounds great, right? We can identify it, except we've just identified it's a really bad guy. Then when we go to try and kill it, you break up that cell, right? When those pieces break up, when that lipopolysaccharide breaks up, the lipid A component is a toxin for us. It is toxic. It's what we call an endotoxin. And so when they do a blood test, right, and they see you've got this toxin, this lipid A in your bloodstream, this is very bad news, right? Because you've got a serious gram-negative infection that as your body, or even if we give you antibiotics, and this is why we have to be very careful with antibiotics for gram-negative bacteria, because if we kill too many too quickly, we're going to kill you by releasing too much toxin too quickly, right? That's why gram-negative infections are scary, why gram-negative infections in your blood can be extremely life-threatening, right? Because now it's traveling system-wide. Okay, so now that I've scared the crap out of you, go have a good day.